Hi everyone. Uh, this video is going to be going over the answers to my practice midterm because we're not going to have time during the LA review session because we're going to be going over the professor's practice midterm and there's no in-depth answer key, so you guys get to hear me talk for the next hour and a half going through my practice midterm. Congratulations. Sorry, it's past midnight and the coffee did not work, so we're having the time of our life. Okay, so we start off with this first question. It says, examine the following reaction. What type of reactant would characterize reagent A? And the key here is looking what's inside this box. Here we see a disulfide linkage, which you should be thinking tertiary or quaternary protein structure. And we see us going from a sulfur-sulfur bond to sulfur-hydrogen bonds. So we've added in two covalent bonds that involve a hydrogen. Okay, this is similar to our redox tower, where on our redox tower you may see something that's written like that. Okay, so we can actually oxidize and reduce sulfur compounds, the oxidized form being an SS bond and the reduced form being an SH bond. So what we see in this box is we're going from oxidized to reduced. In other words, this reaction here is a reduction. Well, remember, redox. It's not read and or maybe ox, it's redox. They always happen together. So if we have a reduction reaction taking place, that means reagent A must be getting oxidized. Okay, because that's the only other thing happening. So reagent A is causing the reduction of something else, making reagent A the reducing agent. Okay, so automatically we can cancel out A and B. We know it's a reducing agent. And now in order for a reducing agent to work, it must be in its reduced state. Remember, the reducing agent is acting as the electron donor. Okay? In order to be a donor, you must have the things that you're going to donate. So in order to be a reducing agent, you must have the electrons that you are then donating to something else. Okay? So it must be in its reduced state. It must have the electrons. Question 2. Molecules what are involved in glycolysis? Molecules what are involved in the fermentation reactions discussed in class? This is kind of an application, sort of. What we see here is we see an NAD molecule. It's technically NAD+. We can see the positive charge there. So this is NAD+, and NAD+ is involved in glycolysis. You know it's involved in the link reaction, which is pyruvate dehydrogenase. It's involved in fermentation. It's involved in the TCA cycle, and it's involved in the ETC. The second one is CoAsh. I think it's technically, I'm going to write it like that, CoAsh. Okay, so we can see it's derived from a nucleotide, and we can see this SH group on the end. Okay, so that's the SH. Notice here with NAD+, it's a dinucleotide. You may also see this written as ADP ribose connected to this molecule here, or not molecule, but that functional group, which is crap, I can't think of the name of what that's called, but just stick with an aromatic ring, I don't know. Versus CoAsh has this large additional group in addition to two phosphates a sugar, and a nitrogenous base, and a final phosphate. And then finally, we have sugar, nitrogenous base, 2 phosphate groups. This is ADP. Okay, so CoAsh is involved in the link reaction, which is the one that takes pyruvate. I don't think this is explicitly stated. The link is going from pyruvate to acetyl-CoA. Okay, so this is involved in the link reaction, and you'll also see CoA involved in the TCA cycle. Okay. Finally, ADP. This is involved in glycolysis. TCA cycle. And instead of the ETC, let's be even more specific and let's say chemiosmosis. I'll say oxidative phosphorylation, because that is the most specific term we can use. Phosphorylation. Okay, so we are just talking about glycolysis and fermentation. So NAD plus and ADP are involved in glycolysis, so that means we should have 1 and 3. And then fermentation, we just see NAD plus. Okay, so that gives us answer choice A. 3, anaerobically respiring. Very quick note, aerobic respiration, our TEA, terminal electron acceptor, is O2. Is in an environment containing excess glucose. For the purpose of this question, assume that there's enough glucose in the environment to sustain this single-celled organism forever. Cool. When the oxygen concentration in the environment drops to 0, so what we're saying is no terminal electron acceptor. What phenomenon would we expect to see? Well, if we have our ETC, we have complex 1, coenzyme Q, complex 3, cytochrome C, and then complex 4, aka cytochrome C oxidase, NADH going to NAD plus, and then we have a bunch of redox reactions as this electron is transferred through. Finally, to complex 4, we have O2 going to H2O, okay, being reduced. And at the complexes, we have H plus being shot across. And then this can be coupled to ATP synthase, which ironically, the diagram is down here. I could have used that. Oh well. Okay. So we have no terminal electron acceptor, which means this does not exist. So this reaction between complex 4 and O2 does not exist, which means that the electrons are going to get stuck on complex 4. If complex 4 gets stuck with electrons, it is unable to oxidize cytochrome C. If it's unable to oxidize cytochrome C gets stuck with electrons, which means it cannot oxidize complex 3, which means complex 3 gets stuck with electrons. Complex 3 gets stuck with electrons, it can no longer oxidize coenzyme Q. Remember, because in order for there to be a redox reaction, you have to have something that's reduced plus something that's oxidized in the oxidized reduced state and oxidized state, switching to the oxidized state, this guy, reduced, right? So you need to have both a oxidation reaction and a reduction. And so if we had, at the end, right, we have complex 4 in its reduced state plus cytochrome C in its reduced state, we have 2 reduced things. So that we'd have 2 oxidation half reactions, or we'd have an oxidation half reaction and then something that's reduced is also then accepting electrons, which doesn't work, right? So that would not be possible. Okay. So the electrons back up until we get to the point where complex 1 can no longer oxidize NADH, and so our NADH concentration in the cell will start to increase, which means our NAD plus concentration will decrease, and as we learned, what will happen is this will trigger fermentation. So with fermentation triggered, our sole ATP source will then be glycolysis. Because remember, fermentation does not generate any ATP. For glycolysis, you get that net gain of 2 ATP, and fermentation is taking that NADH and converting it back to NAD+, which then can go back into glycolysis. Okay. So, let's see. There will be no change as oxygen is not the final electron acceptor, and thus the loss of it in the environment will have no impact on an aerobically respiring organism. This is false. An aerobically respiring organism does use the terminal electron acceptor as oxygen. So, you will expect a change. Glucose concentration outside the cell will decrease more rapidly than when oxygen concentration was high. We'll get to this in a second. Okay. C, the ETC will work faster to maintain the ATP levels required by the cell that would normally be produced by aerobic respiration. Well, here, right, our ETC broke down. So our ETC stops. No more oxidative phosphorylation. So our ETC wouldn't work faster because it's stopped. So that doesn't make sense. In D, the cell will switch from non-cyclic to cyclic photosynthesis to produce more ATP as opposed to producing NADPH for the Calvin cycle. Well, that would be valid if this single-celled organism could perform photosynthesis. But nowhere in this question did it say that the cell could perform photosynthesis. Like, humans can't perform photosynthesis, as cool as that would be, right? We can't do that. And so D could not be true because we have, well, I mean, I guess it could be true, but we have no idea if this cell does photosynthesis. Okay. So revisiting B, if glycolysis is our sole source of ATP, that means that the rate of glycolysis will need to increase. Why? Well, remember, without the TCA cycle, without the ETC, you're losing out on a bunch of ATP. As your sole source of ATP, glycolysis is carrying the entire energy needs of the cell on its back right now. And so that means you're going to need this to increase in rate. So that way you can at least get your ATP, your little bit of ATP out. Because it's either you get 2 ATP out per glucose or you get none. I'd personally choose the 2 ATP because none means I'm dead. Okay. So now by increasing the rate of glycolysis, I'm increasing the rate at which I get this 2 ATP. It's not much, but it's better than nothing. Okay, so if the rate of glycolysis is increasing, well, glycolysis requires that glucose enters the cell, right? So glucose goes outside the cell to inside the cell. And then from here, it goes into glycolysis. Oh, hello. So if we need this to speed up, we need this reaction to also speed up to keep up with our glycolysis needs. Okay, so the rate at which glucose is then entering the cell increases. So if the rate that it's entering the cell increases, Well, that glucose had to come from somewhere. So if it's going from outside the cell to inside the cell, the concentration outside the cell will start to decrease. And it will decrease much more rapidly than when you got a lot of ATP out. Because when glycolysis is running much more slowly, aka when the cell is able to extract a lot of energy from the glucose, the rate at which glucose enters the cell does not need to be very, very high. Versus as when you're only getting 2 ATP for glucose. When you need a lot of glucose entering the cell in order to keep up with the rest of the cellular needs. Okay. So it has to be B, because it's saying is glucose is entering the cell at a faster rate, and we can't create nor destroy matter. So if it's increasing at a faster rate, its source must be decreasing at a faster rate. Okay. So next we have questions 4 through 7. We'll use this diagram. You're welcome for putting all the questions in the diagram on the same page. I'm kidding. So this is basically exactly what I drew up above. And we'll talk about some applications of this. Okay, the molecule shown to the right is dinitrophenol. Cool. You might also see it as written as 2,4-dinitrophenol. Dinitrophenol is just the colloquial term, but technically the IUPAC name would be 2,4-dinitrophenol if you take an OChem. When inserted into the inner mitochondrial membrane of humans, it is observed that ATPase function, aka this guy, is greatly reduced, but lactic acid concentration does not increase. So what it's saying is when we stick dinitrophenol inside the membrane, ATP function more or less stops. Okay, so this is inhibited, but we do not see any lactic acid concentration increasing, meaning that fermentation is not triggered. So if fermentation is not triggered, that must mean that the ETC is operating normally. Because if the ETC stops, the cell would move on to fermentation. But the ETC is operating normally because we do not see fermentation being triggered. So what this tells us is that all this electron flow is happening just fine. There's no issues there. Okay, so then we have to ask ourselves, well, why would ATPase function, or ATP synthase function, be greatly inhibited? Well, it gets into ATPase function is due to this H plus gradient. Okay, so when we have a low pH here, aka high H plus concentration, and a high pH down here at location A, we have a spontaneous flow of H plus going down its concentration gradient, right? That's chemiosmosis, which provides the energy necessary for ATP synthesis. And so if ATPase function is greatly decreased or not happening at all, what we're saying is we do not have a very spontaneous flow of H plus. Well, how could we get that? And that would be if these pHs are roughly equal. So if the pH of B is approximately equal to A, right, that's saying that their H plus concentrations are about equal, meaning that neither side is really favored, right? So going from location B to location A, this is no longer a very spontaneous process, right? The reverse direction is equally as likely. And that would account for the ATPase function being severely reduced. Okay, so we now have figured out how we can get ATPase function to be very reduced. Now we just need to say, okay, how do we go from a functioning ETC that's generating a PMF, a proton gradient, to not having a proton gradient? Well, the answer is what dinitrophenol is doing. So when it's, oh my gosh, I just realized I put DNA here. Getting ahead of myself. When we introduce dinitrophenol, what it's doing is it's breaking apart the membrane such that H plus can travel right through. When this happens, right, the cell is putting all this energy into moving H plus across the membrane. And so very momentarily, you have a little bit higher concentration of H plus at location B than you do at location A. But because you have this pathway for the H plus to flow through with very low activation energy, what's happening is the H plus is just going right back through. So we are doing active transport to get it over, and then it goes right back through. And it's bypassing ATPase function, right? We're not able to establish this proton motive force that's exergonic, that can power ATP synthase function. Okay. And so the ETC is just fine, right? There's no issues there. It's just that the product of the ETC, this H plus gradient, is getting destroyed by dinitrophenol by allowing the H plus concentrations to equilibrate without ATP synthase's function. So our answer choice is D. A, it allosterically binds to complex 4, preventing the reduction of O2 to H2O and halting the ETC. If it halted the ETC, we would see lactic acid concentrations increasing because we would go to fermentation. So it's not A. B, it prevents complex 2 from oxidizing FADH2 to FAD, stopping the movement of electrons within the ETC. So the problem, well, this one gets more into, how should I say? You would still have electron flow from NADH and NAD plus through complex 1, right? So these goes like that versus FADH2 just goes like that. So you would still have electron flow via NADH. The issue is all your FADH2 would build up in the cell. And once it builds up in the cell, you have no FAD. And if you run out of FAD, the TCA cycle would stop. If you run out of FAD. So this says the TCA stops, which would likely trigger fermentation. Or introducing another pathway to oxidize this FADH2. So kind of multiple reasons why this is wrong. It doesn't actually stop the movement of electrons because you have NADH that can still be oxidized just fine. Okay, so it's not B. C, it creates a novel pathway between locations C and B, allowing protons to travel through the membrane and preventing the formation of an electrochemical gradient. Well, if this were true, H plus goes through here. Okay, well, location C and location B could have roughly the same pH. But we could still end up with this pH being lower than location A's. So even though location B and location C have the same pH, they could still be lower than location A, meaning that location B and C have a higher H plus concentration than location A, which means you would still be able to get ATPase function or ATP synthase function. So C doesn't work. D, we already said, that's the answer, right? And E, it inhibits the enzyme that converts pyruvate into acetyl-CoA. Well, if this is true, aka pyruvate dehydrogenase, that's the enzyme it's referring to, without this reaction taking place, so pyruvate dehydrogenase, PDH, So what this is saying is dinitrophenol is inhibiting PDH. If this were true, our pyruvate would then be undergoing fermentation. Okay, so it would go through fermentation. But we said fermentation doesn't occur. So E doesn't work. Okay, so this is just a question of understanding how the ETC fits in with the rest of cellular respiration and metabolism in general. Okay, question 5. Cyanide, when introduced into a cell, binds to and inhibits complex 4, also called cytochrome C oxidase. What would happen to the pH difference between location B and location A after cyanide has bound complex 4? So, when cyanide binds, complex 4, and what this does is it prevents electron transfer. Transfer to O2. Okay. So when this happens, our electrons build up in the cell because cytochrome C oxidase, or complex 4, is unable to pass its electrons onto O2, which means electrons build up in ETC. And as we talked about earlier, when you have this electron buildup, the ETC stops. And when the ETC stops, we stop getting this H plus being pumped across, right? So no more H plus pumped across, which means you have no force or no external input maintaining this electrochemical gradient. And so after time, the H plus concentrations at location B and location A should even out because you're no longer pumping protons across via active transport into location B to keep that concentration of H plus high. So eventually the H plus concentration, so the concentration of H plus at A will equal the concentration of H plus at B because you're no longer artificially increasing the H plus concentration at B. So they'll equilibrate, or they'll reach equilibrium, and we'll see that the pH should be approximately equal. Which of the following statements correctly describes a possible redox relationship from the diagram? ATPase has a greater reduction potential than complex 1. Is ATPase involved in electron transfer? Transfer? No. So ATPase is not involved in electron transfer. Right? What it's doing is it's taking the H plus, undergoing chemiosmosis to bind ADP and P sub I to make ATP. It's not involved in any electron transfer. So ATP synthase is not part of the ETC. They're coupled together. They can be coupled together. B, complex 1 has a greater reduction potential than ATPase. Same thing, right? ATPase is not involved in redox. O2 has a greater reduction potential than complex 1. Remember, we want our delta E to be positive. So if we take... Actually, I should write this out. So our delta G is equal to negative NF delta E. For delta G to be negative, we need delta E to be positive. So for our delta E to be positive, what we need is the acceptor, or the final, minus the donor, or the initial, to be greater than 0. So if O2 has a greater reduction potential than complex 1, what it's saying is the reduction potential of O2 minus the reduction potential of complex 1, this is greater than this value. Okay, so if you have something minus something that's smaller, let's give arbitrary numbers, 5 minus 4, right, because 4 is less than 5. you get a number that's greater than 1. Okay. I don't remember what oxygen is 0.82 volts. I don't remember what complex 1 is. But so we need this to be higher in terms of reduction potential. That way it can steal electrons. Okay. So C is true. D, complex 1 has a greater reduction potential than O2. If this were true, this term here would be greater than this guy. So you have something minus something that's bigger. You'd end up with a delta E less than 0, meaning that it's non-spontaneous. Okay. And then finally, E, NADH is the initial electron donor, and complex 4 is the terminal electron acceptor. And note that initial and terminal refer to the entire ETC. This is an enticing answer, right? Because NADH is the initial electron donor, but complex 4 is not the terminal electron acceptor. Remember, that's O2. O2 is where the electrons are going. It is like the electron dump. So complex 4 is not the terminal electron acceptor. O2 is the terminal electron acceptor. Okay. So question 7, focusing on the reaction between NADH and complex 1. So what we can say is NADH plus complex 1 goes to NAD plus complex 1. And we need to decide, is complex 1 donating or accepting? And is it reduced or oxidized? Well, if we use our rules, here we have NADH going to NAD plus. In our minds, we should be thinking this is an oxidation reaction. NADH is donating its electrons to something else and be undergoing an oxidation. So NADH is donating the electrons. We can cross out C and D because it's telling us that NADH is accepting the electrons. So that's not true, right? It's not accepting, it's donating. Okay, and now we need to decide, excuse me, is complex 1 in an oxidized or reduced state? Well, if our complex 1 is taking part in this redox reaction and we have an oxidation, we must also have a reduction, which means complex 1 is undergoing a reduction reaction, which means as a product, it must be reduced, right? It's gaining these electrons. In order to gain electrons, it needs to be in a state where it can gain those electrons, meaning its oxidized state. So the reduction part is going from oxidation or an oxidized state to a reduced state. And we know that by knowing what happens to our NAD molecule. In this case, NADH is going to NAD plus, which is an oxidation, which means we need to have a reduction elsewhere. So the answer is A. Okay. Examine the reaction. AX plus NAD plus goes to AY plus NADH. Delta G is less than 0. X and Y are labels that should read respectively. This is actually very similar to the previous question, except instead of focusing on donating and accepting electrons, I'm just asking you about what should be here. Okay. So we see NAD plus going to NADH. I see that my reaction does occur. And so if our NAD plus is going to NADH, this is a reduction half reaction. In order for reduction to take place, we must have an oxidation reaction, which means AX to AY, we're going from a reduced state to an oxidized state. Remember, that's an oxidation reaction. We're losing the electrons. In order for something to lose electrons, it needs to have those electrons in the first place. It needs to be reduced. And so the X here corresponds to the reduced state, and the Y here corresponds to the oxidized state. So answer choice 8. All right, which of the following statements most accurately describes kinesins and microtubules? Remember, microtubules are composed of tubulin. Look like this. Okay. And then on top, we have the kinesin that then is attached to some blob that is taking. Okay. And this guy will move. So the microtubules are structural proteins versus the kinesin, which is the motor protein. But note that they're both made of proteins. Microtubules, are made of tubulin, of which there's both the alpha and beta conformation of tubulin. Okay. Versus kinesins, which are your motor protein. Now, microtubules will also use dyneins. Okay. Quick tangent on this is the microfilaments, which are made up of actin, which is still a protein, and it uses the myosin motor protein. Okay, so to answer this, both are made of proteins. However, kinesins are motor and microtubules are structural proteins. We know that's true. Kinesins convert mechanical energy to chemical energy, which it uses to travel along microtubules. Close, but it's the opposite. Remember, as kinesins travel, what they're doing is they're hydrolyzing ATP. So as kinesins travel, they're taking ATP plus H2O, ADP plus P sub I plus energy. And this energy can be used to move. Okay, so we're converting chemical energy, right? The energy inside of ATP to mechanical. So it's not B. C, kinesins and microtubules both require binding to ATP to function. This is false. Kinesins do, but microtubules bind to GTP. Okay, well, we know B and C are wrong, so it can't be all of them. And we know B is wrong, so it can't be E, which makes our answer A. Okay. Question 10. Dr. Fashadi asked you to remember 3 big key ideas regarding cytoskeleton fibers. Those are, they are composed of monomers. Remember the cytoskeleton. You have tubulin, which has a monomer, right? The alpha and beta dimers. So the alpha and beta tubulin form a dimer, which itself is the monomer for microtubules. And actin is the monomer for microfilaments. They're unsymmetrical. Remember how he showed in lecture that they have polarity. You have a positive end of the microtubule and a negative end of the microtubule. Actually, I think it should be the other way. Yes. We have the positive end and the negative end of the microtubule, right? So there's a polarity. It has to do with what face of the protein is showing out. Okay. And then you'll also, probably in your upper div classes at some point, you may learn more about tubulin and how the different sides correspond to what type of, well, not GTP. Basically, on the positive end of GTP, the beta tubulin is bound to GTP. Sorry, the positive end of the microtubule, beta tubulin is bound to GTP, and on the negative end, it's bound to GDP, which is why the negative end can depolymerize pretty quickly. We don't talk about that with this professor, so don't worry about that, but that's something you may come across in the future. But it has to do with their unsymmetrical. And then finally, they're dynamic. Oftentimes, right, with these still drawings, they're very still. They're very undynamic. But we want you to remember that these are constantly polymerizing, depolymerizing. Things are constantly moving, right? It's in this state of movement. It's very, very dynamic. Okay, so this is basically a memorization question that I thought was important. Okay, in humans, amylase is secreted from the salivary glands in high amounts to digest starch and glycogen in food, in comparison to a cell that is not involved in the secretion of enzymes. That's the key. The salivary gland cells will have a larger amount of which organelle. So it's asking, this salivary gland does a bunch of secretion of enzymes. So if you're doing a bunch of secretions of enzymes, you should have a lot of an organelle that is responsible for the secretion of enzymes. And the organelle that's responsible for that is the Golgi apparatus. So the nucleus, it's not the nucleus because the nucleus is for DNA storage. Chloroplast is for photosynthesis. The Golgi apparatus is for protein packaging for outside the cell. AKA secretion. And then the ribosome is responsible for peptide synthesis, which once we hit transcription and translation, we'll talk about this more. So this is translation, which we will talk about maybe in like 2 weeks. I don't know. I don't know what week it is. It all just goes over my head. Anyways, question 12. Without knowing another piece of information regarding the overall reversible reaction shown below. So if it's reversible, that tells us one of the directions is favorable. What can we say for certain about the reaction? So one of these directions is favorable. Do I know which one is favorable? I have no idea because it was not stated to me. So to say A to B is endergonic, how do I know that A to B is endergonic? Right? There's nothing that tells me that A to B is endergonic. B to A could also be endergonic. A to B could be exergonic. I don't know. What this is trying to teach you is to not get sucked into this idea that NAD plus to NADH is very, or what I often hear from students is, oh, NAD plus to NADH, this reduction is endergonic. This is not true. Because what this is saying is NAD plus to NADH is a reduction reaction. If it's endergonic, means that if I supply energy to this, it will occur, but then I have reduction occurring without an oxidation reaction. And remember, it's redox, not read and maybe ox. It's redox. It always happens together. So saying NAD plus to NADH is endergonic is wrong. We can only say whether or not a redox reaction is exergonic or endergonic. A half reaction is neither exergonic or endergonic. Okay? So I don't know. Perhaps A plus NAD plus going to B plus NADH has a delta E greater than 0, but it could very well, just as well, have a delta E less than 0. I have no idea. There's nothing that tells me which side is favored. So A is wrong. D, the overall reaction to the right is exergonic. I have no idea. A is the reduced state. B is the oxidized state. Now, what we do know is one of these, because it's a reversible reaction, we know one of these reactions, or one of these, the reverse of the forward reaction has a delta G less than 0, which means one of the ways does work spontaneously, meaning that you have a valid redox reaction here. So this is telling you you do have a valid redox reaction. Note that valid redox reaction, reaction. This does not mean exergonic or endergonic. It just means it's valid and it's possible. It may be spontaneous, it may be not spontaneous. We don't know. But we know it's valid. And we know one way, either the forward or the reverse, is spontaneous and the other one is non-spontaneous. Okay, so if you have a valid redox reaction and we see NAD plus and NADH involved, so if I just look at the forward reaction as written from left to right, I see NAD plus going to NADH, that's a reduction reaction. That must mean A to B is an oxidation reaction. So A is going from a reduced state to an oxidized state in B. Okay. Like I said, I don't know which direction is spontaneous or not. I just know one of them is, which tells me that we have a valid redox reaction, which means that my A state must be the reduced state, because that's the only way to have a valid redox reaction. So B works. I know D doesn't work because the overall reaction to the right is exergonic. I have no idea if that's true. So B must be our answer. And then C, A is the oxidizing agent. NAD plus is the reducing agent. Well, in order for this to be true, that means A is causing the oxidation of NAD plus, but we see NAD plus is getting reduced. So C doesn't work. Okay, 13. A prokaryotic cell utilizes cellular respiration as its source of cellular energy. Where does the cell pump its H plus to establish the PMF required for respiratory ETC? Remember, we're dealing with a prokaryotic cell. Does a prokaryotic cell have membrane-bound organelles? No. So, can it be pumped into the intermembrane space of the mitochondria? If you're a eukaryote, yeah. But for a prokaryote, it doesn't have a mitochondria. So it can't pump into a place that doesn't exist. B, outside the cytoplasmic membrane, that's the only membrane you have available, right? For those prokaryotes, they only have that cytoplasmic membrane. Now, once you take Mike 102, you'll learn about this, how it actually works, and it's pumped into a place called the periplasm space. But for our purposes, we don't know that. All we know is there's only one membrane, one plasma membrane. And so it has to be pumped outside of that. C, into the matrix of the mitochondria. Mitochondria doesn't exist. D, inside the cytoplasmic membrane. Well, remember, that's where all our processes are taking place, right? That's where NADH is getting oxidized to NAD+. And we're pumping protons on the other side. So either we have to move our NADH to the outside the cell membrane to pump the protons in, which seems horribly inefficient, right? So we have to, in order to pump the protons into the cell, we have to move all our NADH outside the cell. And that would also mean our ATP synthesis would occur outside the cell, right? Our ATP synthesis would occur like here if protons were getting pumped into the cell to establish the gradient. So that doesn't make sense. Okay. For 14, which of the following is not a function of the cytoskeleton? Cellular structure and rigidity. This is a function of the cytoskeleton. Protein packaging. We already learned that's the role of the Golgi apparatus. Okay. The Golgi apparatus is not a part of the cytoskeleton. Also, I think technically the ER as well. Oh, I don't remember. Oh, well. We know A is a function of the cytoskeleton. And so it's not a possible answer. Cellular movement. The cilia and the flagella are all made up of cytoskeleton elements. Intramolecular macromolecule transport. Microtubules. Function of the cytoskeleton. Cellular organization of organelles. a point of the cytoskeleton, right? The cytoskeleton acts as this compartmentalization. It can organize the organelles to keep things separate, so keep the reactions separate, and also provide a pathway for you to go between your organelles. Okay? So it organizes the organelles inside the cell to keep things very controlled, very organized. Prevents, cross-contamination between reactions, keeps everything doing their job. And it will also help, how should I say? What's the term? Tether things. So the cytoskeleton will help tether organelles and other things to the cell instead of just freely floating. Okay. Questions 15 and 18 will use this metabolic pathway. 15 says, which of the following would be most appropriate to describe the half reaction Y to Z? So in this reaction, we see metabolite 1 plus Y going to metabolite 2 and Z. And what I'm seeing, the difference between metabolite 1 and metabolite 2 is the addition of this phosphate group. So I needed to get that phosphate group from somewhere. So somewhere in Y, I needed a phosphate group. But remember, I'm also making a very high energy bond. And so I need energy to do that. Well, so that energy must have come from somewhere, because going from no phosphate group to a phosphate group, we know that M1 to M2 is endergonic, right? We're adding a phosphate group on. We know that requires energy. So that means Y to Z has to, 1, provide a phosphate group to be attached, and 2, it needs to provide the energy to do such, meaning that its delta G needs to be less than 0. Well, what reaction can provide a phosphate group and provide energy? Well, that's ATP to ADP, right? So when we take this, we hydrolyze it, we'll end up with ADP plus P sub I plus energy release. So there we go. P sub I, energy, we've just satisfied the 2 requirements we needed out of our half reaction. Okay, so that means Y is equal to ATP and Z is equal to ADP. Question 16. Reaction 3 is best categorized as, let's see, we're adding a phosphate group on, but we also see NAD plus going to NADH. That's a reduction reaction, which means M3 to M4 must be an oxidation. So it's a redox reaction. Okay, so our answer is redox. It's not hydrolysis. We're not breaking apart any phosphoanhydride bonds. It's not a condensation because we're not making any phosphoanhydride bonds. We're just sticking a phosphate group on. Answer choice D says uncatalyzed. The enzyme's right there. So it is catalyzed. And E, substrate level phosphorylation, we see a phosphate group getting attached to a substrate, but that's not substrate level phosphorylation. Remember, substrate level phosphorylation is when you have a substrate with a phosphate group plus ADP going to that substrate plus ATP, where the substrate loses its phosphate group. This is substrate level phosphorylation. Okay, 17. A cell that wants to maintain high favorability of reaction 3 should, and this is, I probably should have wrote this up here, but whatever. M1 and M4 are the labels for the molecules they are next to. We want to maintain high favorability of reaction 3, meaning we want to make this one make sure it has a delta G that's less than 0. How can we do this? Well, as we learned on Wednesday, which the day of recording this was yesterday, we learned that if we keep reactant concentrations low and product concentrations high, we will favor the reverse reaction. And so if we keep reactant concentrations high and product concentrations low, we favor the forward reaction. And remember, favorability. If we increase the favorability of a reaction, we're decreasing its delta G, right? Its delta G is becoming more negative. So this is just Le Chatelier's principle. You increase the reactant concentrations and decrease the product concentrations. You shift the reaction to favor more products, aka you're making that forward reaction more favorable. In other words, you're making it spontaneous and you're decreasing its delta G. So to do this, we need to keep reactant concentrations high and product concentrations low. And so keeping the concentrations of M3 high, aka the reactant, and M4, the product, low. Now, if we were to keep the concentrations of M2 low and M3 low, we'd be having very low reactant concentrations, so we'd be favoring the reverse reaction. We'd make it less favorable for reaction 3 to proceed forward. For C, keeping the concentrations of M1 high and M4 high, if we keep the concentrations of M1 high, we may increase the amount of M2 we get, which would then increase the amount of M3 we would get. But our issue is with the high M4, right, we're increasing our product concentration, which means we would be disfavoring more product concentration. So the issue is this guy here. D, keep the concentrations of M2 low and M4 high. It's the same thing. If you have low M2, you'd be favoring the reverse reaction. So M3 to M2. And by doing that, you're getting even less of M3, which means you're favoring this, the reverse reaction, and you're increasing M4, which also favors the reverse reaction. So D would also make it less favorable in the forward direction. Okay, 18. Suppose that a new enzyme was introduced to the cell, enzyme 4, that catalyzes a new reaction between M4 and ADP to generate M5 and ATP. For this reaction to proceed spontaneously as described above, so what we're saying is M4 plus ADP goes to M5 plus ATP. Oh my gosh. And my delta G is less than 0. I need the delta G's of this when we add together. So M4 to M5 with delta G1 and ADP to ATP, delta G2, I need the sum of this, so delta G1 plus delta G2, to be less than 0. For this to be the case, well, let's see, ADP to ATP, that's an endergonic reaction, which means this delta G is going to be greater than 0. So for the total one to be less than 0, I need M4 to M5 to have a delta G less than 0. Okay, but now I need to make sure that this guy, delta G1, releases more energy. So M4 to M5 releases more energy than is required to generate ATP from ADP. In other words, the magnitude of this delta G needs to be greater than the magnitude of delta G2. Another way to say that is delta G1 is more negative than delta G2 is positive, which corresponds to answer choice C. Okay, I'm going to start to keep this up because it's 1:30 and my throat is dying because I've spoken for like 6 hours today while tutoring. Okay. Using the diagram to the right, which answer choice contains the correct set of compounds in their more reduced forms? All this question is asking you of the molecules present here, which ones are in their reduced forms? Remember, we always search for the molecules we know. We see NADH and NAD+. Okay, in order for NADH to be reduced here, so this is undergoing a reduction half reaction, or reduction reaction. That means malate to oxaloacetate needs to be an oxidation reaction. Okay, so NAD plus is in its oxidized form. NADH is in its reduced form, which means that malate needs to be reduced in order to be undergoing an oxidation reaction to oxaloacetate, which means that oxaloacetate should be in its oxidized form. Okay, so the compounds that are in their more reduced form would be malate and NADH. Okay, 20. Fermentation is a cellular process in which A, NADH is recycled back to NAD plus by reducing a small, highly abundant molecule which enters the cell for respiration. NADH is recycled back into NAD plus, but this small, highly abundant molecule which enters the cell for respiration is describing the terminal electron acceptor. Right? Something like O2. Fermentation does not require O2. It does not require a terminal electron acceptor. Right? That's for the ETC. So it's not A. B, ATP can be directly generated without having to utilize the ETC. Nope. Fermentation does not produce any ATP. Its job is to recycle NADH into NAD+. C, NAD plus is reduced to NADH to be used elsewhere in the cell. Nope, that's the exact opposite of what fermentation does. D, NADH is oxidized to NAD plus by reducing a molecule produced from a related cellular process where the electrons were harvested. Well, let's see, a related cellular process where the electrons were harvested, that's referring to glycolysis. And NADH is oxidized to NAD plus by reducing a molecule produced from that related cellular process. That's referring to pyruvate. And remember, fermentation, we have pyruvate undergoing a reduction to something like ethanol, and that's coupled to the, well, not coupled, right? Because we do not couple. Well, redox reactions happen together, right? You can't have reduction without oxidation. So you have the reduction reaction of pyruvate to ethanol, and that is occurring with the oxidation reaction of NADH to NAD+. Answer choice is D. E, glucose is completely oxidized to CO2 and is excreted from the cell. That's describing cellular respiration. Right, via the TCA cycle, which does not happen. Okay, we're doing fermentation. Okay, 21. Membrane-bound organelles were an important evolutionary step in the development of modern eukaryotic cells. As a result of these novel compartments, cells gained the ability to more efficiently A, carry out particular cellular processes in highly specific microenvironments. So if you have an organelle, what you can do is you can regulate the environment inside this organelle. So maybe you have a very, very low pH. Whereas if you were to have this in the cytoplasm, it could potentially disrupt a bunch of other proteins, causing the cell to die. But if you keep a very low pH inside this very controlled organelle and have only enzymes in here that require that low pH, well, any enzymes that are outside of this organelle will be unaffected. This is actually how lysosomes operate. Okay, so A is true. B, they separate chemical reactions from each other to prevent interference. If I have a chemical reaction occurring in this organelle and a chemical reaction occurring in this organelle, well, even if they were to use the same reactants and products, they're happening independently of each other, right? You have this spatial separation between the two. You're isolating these processes to make sure there's no crosstalk. C, increased membrane surface area for reactions requiring a membrane. So if we think about cellular respiration in prokaryotes versus eukaryotes, in prokaryotes, cellular respiration, aka a process that requires the electron transport chain, utilizes the plasma membrane, the cellular membrane, in addition to all of its transporters, cell signaling, any cell movement, so cytoskeleton stuff, All of that needs to occur, although prokaryotes don't have a true cytoskeleton. They have it. You'll learn about it in another class. All this stuff needs to occur on the cytoplasmic membrane versus eukaryotes, which can have all their electron transport chains on, or at least the energy harvesting ones, they can have them on the mitochondrial membrane to save space on the cellular membrane, which can be then used for more space for other things such as membrane transporters for getting stuff inside and outside the cell. So they've increased the membrane surface area by splitting what reactions are present on what membrane. So some reactions are going to be present on the mitochondrial membranes versus others that are present on the cellular membranes versus a prokaryote, which has everything on one membrane. So our answer is D. Okay, ATP plus H2O goes to ADP plus P sub I, has a delta G naught prime of negative 7.5 kilocalories per mole. Which of the following reactions could be effectively coupled to the production of ATP from ADP and P sub I? So we're producing ATP from ADP and P sub I. So if going in this direction has a delta G naught prime of negative 7.5 kilocalories per mole, that means ADP plus P sub I going to ATP plus H2O has a delta G naught prime of 7.5 kilocalories. Right? Because we're just doing the opposite reaction, so it should have the same magnitude of delta G, not prime, but different sign. Okay, so which of the following reactions could be effectively coupled? In other words, the delta G, not prime total, is less than 0. Let's see, if I add in reaction 1, I have 7.5 kilocalories plus 4 kilocalories. That's positive. So our delta G naught prime total would be positive, which means it's endergonic, so that doesn't work. So any answer choice with 1 is automatically out. Okay, how about answer choice 2? I have negative 4.5 kilocalories per mole. Well, negative 4.5 kilocalories per mole, oh my God, plus a negative 7.5, positive 7.5 kilocalories per mole. Well, negative 4 plus 7.5 is 3 kilocalories per mole, which is still positive, so that doesn't work. And so any answer choice with that could automatically get taken out, and so that only leaves us with answer choice B. But B says answer choices 3 and 5. So let's see, negative 10 kilocalories plus 7.5, that's negative 2, which is negative, that works. 6 plus 7.5 is 13.5, which is positive, doesn't work. And then finally, I didn't mean to do that. There we go. Then we have negative 8.7 kilocalories per mole plus 7.5, which is negative 1.2 kilocalories, which is negative. So they would be effectively coupled. God, my throat is dying. Okay. Let's not talk about this one, 23, because photosynthesis is no longer on your exam. So we'll revisit it for midterm 3. 24, using the table to the right, HSO4 minus plus NAD plus 2H plus goes to SO4 2 minus plus NADH. When we're going through, we need to make sure that we have a reduced form and an oxidized form. HSO4, according to this table, is reduced. NAD plus is oxidized. So we have a reduced state and an oxidized state going to an oxidized state plus a reduced state. So we can consider whether or not this is exergonic or endergonic, right? So it is possible because we have something reduced and something oxidized. Okay, very quickly, I can actually cancel out answer choice D because this says exergonic and non-spontaneous, which they can't be both, right? Exergonic means to be spontaneous. So it can't be D just by logic. Okay. And now we need to calculate the delta E's. So E acceptor minus E donor my acceptor is NAD+, so I have negative 0.32, minus my E donor, which is HSO4, so negative 0.52, which is negative 0.32 minus a negative, which is the same thing as a positive, 0.52, which is equal to 0.2 volts. So my delta E is equal to 0.2 volts, which is greater than 0, which means my delta G, my delta E is positive, which means my delta G should be negative. My delta G is negative, that means it's exergonic and spontaneous. Okay, using the same table to the right, which of the following reactions would not occur without the external input of free energy? So that means if I provide external input of free energy, I can get a reaction to occur. Meaning, this is asking us for the endergonic. Endergonic reaction. Let's see. 4H plus NAD plus goes to H2 plus NADH. We have an oxidized thing, 2H plus, and NAD plus. We have an oxidized plus an oxidized form. That's not endergonic. That's not exergonic. That's not physically possible. Because you have two oxidized things. Which one's going to be the electron donor? Neither of them. B, H2 plus NADPH. Well, NADPH is reduced. So is H2. So you have two reduced forms. Which one's going to accept the electrons? Neither of them. Okay, now we have glucose plus 12 NAD plus goes to 12 CO2 plus 12. Also, there should be a 12 there. Oh, well. 12 NAD plus goes to 12 CO2 plus 12 NADPH. Well, we have glucose, which has a E value of negative 0.43. And it's going to be the donor. So we have a reduced form and an oxidized form. So that means it is possible now, because glucose is reduced. NADP plus is oxidized. So it is possible. So we'll do acceptor minus donor. The acceptor is NADP plus, so negative 0.32, minus the donor, which is glucose, negative 0.43. It's the same thing as negative 0.32 plus 0.43, which is 0.09 volts, which is greater than 0, which means my delta G is less than 0. Okay? Which means that it will occur regardless of the external input of free energy. Right? This is an exergonic reaction. So C, while exergonic, it does not answer our question because we're looking for the endergonic reaction. Okay. Now, for E, there's no amount of external energy input that can cause a non-spontaneous redox reaction to occur spontaneously. That's wrong, right? Non-spontaneous reactions means if you supply the free energy input, you can get that reaction to occur. Now, if we do D, which by process of elimination, it has to be the correct answer, we have NADPH, which is reduced, and SO42 minus, which is oxidized. So you have a valid redox reaction. You have a reduced thing and an oxidized thing. Our donor is NADPH. Our acceptor is SO42 minus. So our acceptor is negative 0.52 minus our donor, negative 0.32. Same thing as negative 0.52 plus 0.32, which is equal to negative 0.2 volts. So our delta E is less than 0, which means our delta G will be greater than 0, which means it's non-spontaneous. Okay, 26. Allosteric inhibitors are best described as, well, remember, allosteric means it binds. Come on. Oh, God. Please stop. Can you just... Thank you. It binds to a site other than the active site. Okay? So we're not binding to the active site. So A, molecules that bind to the active site that allow an enzyme to catalyze a reaction. I just said allosteric does not bind to the active site. So A is wrong. B, molecules that bind to the active site. Nope. Wrong. C, molecules that bind to an active site, to a site other than the active site. Okay, so that matches our definition. That allow an enzyme to catalyze a reaction. Well, that would be true of an allosteric activator, right? And something that allows an enzyme to catalyze a reaction describes an activator. It's turning on an enzyme. We're asking about an inhibitor. So an inhibitor turns off an enzyme / protein. Okay. So this part of the, what's it called? The answer choice is wrong. E, any molecule that binds to the enzyme to influence the enzyme's function. This is wrong because competitive inhibitors and activators will both bind to the enzyme and influence the enzyme's function. Well, kind of. Not really. More so, the better explanation for this one is inhibitors, right? So inhibitors are strictly talking about turning off the enzyme, whereas this one's just saying any influence on the function, turning it on or off. So that's wrong because we're specifically talking about inhibitors. So that takes us to answer choice D, molecules that bind to a site other than the active site that prevent an enzyme from catalyzing a reaction. That hits the allosteric part and the inhibitor part. Oh my gosh. All right, 27. Cancer is caused by the deregulation of cell checkpoints within the cell cycle. One of these checkpoints is regulated by tumor suppression protein p53. In most types of cancer, p53 is mutated, which also has the side effect of preventing the synthesis and assembly of mitochondrial proteins essential to the passage of electrons to O2. How would we expect cancer cells to behave compared to normal cells? What this is saying is that our complex 4 is unable to pass electrons onto O2. It does the same thing as cyanide, right? So remember we talked about earlier, when we introduce cyanide, where is it? Here. We start blocking the ETC from functioning, right? The electrons build up in the ETC and the ETC stops. So in this case, right, what we're doing is we're preventing O2 from getting these electrons. Electrons build up, and the ETC stops. If ETC stops, the cell is going to trigger fermentation. So let's see, the production of lactic acid / ethanol would be equal to that to a normal cell? No, because we've now triggered fermentation, and a normal cell would likely not be going under fermentation. B, the production of lactic acid and ethanol would be more than that of a normal cell. That's true, right? Because a normal cell would probably not be undergoing fermentation versus our cancer cell is undergoing fermentation. C, the rate of the ETC would increase. Well, the ECT, oh my gosh, it's late. The ETC has stopped, so it should decrease. D, the rate of the ETC would be equal to that of a normal cell. Once again, it wouldn't be equal. It should be less than. It needs to decrease. E, the cancerous cells would become more efficient in harvesting energy from glucose compared to a normal cell. Well, if that were the case, it would be harvesting more ATP from each glucose molecule. But remember, in fermentation, when a cell is fermenting, all of its ATP is coming from glycolysis, which is 2, versus anywhere around like 36-ish ATP when it's undergoing cellular respiration. So fermentation is not efficient. But you do fermentation if you don't have your terminal electron acceptor because it keeps you alive long enough for you to go get access to more of your terminal electron acceptor. So it definitely does not become more efficient. It becomes less efficient. So our answer is B. If you want to read more about this, it's called the Warburg. Hold on. I want to make sure I spelled that correctly. Yep, Warburg effect. It's pretty cool. Warburg effect. I couldn't remember if it was URG or ERG, but it's URG. Okay. 29. Examine the diagram below regarding one of the steps in the overall reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Which of the following bonds, pointed to by red arrows and labeled A through D, is example of a high energy bond? Well, let's see. Here is a bond within an amino acid. This is histidine. So we have a bond within the amino acid itself that does not match our definition of a high energy bond. Right? So it's not A. If we look at B, if we see here, we go from one CH bond to another CH bond. And we see NAD plus going to NADH. So this here, this bond is a redox bond. But this is not a high energy bond. Remember, high energy bonds have to deal with hydrolysis, right? If you hydrolyze them, you get a large release of free energy. That's not the same thing as a redox bond or a bond that engages in redox reactions. So it's not B. D, once again, this is a bond inside of histidine. This is actually an acid, or this is an acidic bond. Once again, not hydrolysis oriented. So it's not D. That leaves us with C. Logically, that has to be our right answer. But we also know that thioesters are high energy. And if we see here, we have a thioester. There's our thioester bond. And we know if we hydrolyze this, so if we stick in H2O, we'll end up like this, plus a lot of energy. Okay, so our thioesters are high energy bonds. Okay. 29. Which of the following reasons would explain why an anaerobically respiring organism would not completely oxidize pyruvate through the TCA cycle in ETC? Anaerobically respiring organisms, aka is TEA, its terminal electron acceptor, is anything except O2. Literally any molecule that is possible of accepting electrons could be the thing here, except for O2. Because O2, if it's the terminal electron acceptor, is aerobic respiration. So, A lack of O2 available to the organism to act as a terminal electron acceptor. An anaerobically respiring organism doesn't use O2 as its terminal electron acceptor, by definition. So it can't be A. And yes, this answer choice is there to trick you if you're reading too quickly. You're welcome. B, to retain carbon for lipid synthesis. Remember how we talked about stuff can go through acetyl-CoA and go into a bunch of other processes. Right? So you can run the TCA cycle in reverse. You can go through acetyl-CoA. You can go through pyruvate. You can convert molecules into each other by going through these central metabolites, such as acetyl-CoA. So we know it's not A, which means D's out. And that leaves B and C. So by going through acetyl-CoA, we can actually retain carbon for lipid synthesis. We can put that acetyl-CoA through lipid synthesis and carbohydrate synthesis. So by stopping at acetyl-CoA, we can put it into lipid synthesis and carbohydrate synthesis. So our answer is E. Okay, 30. Last question. The cartoon diagram to the right shows the immediate phosphorylation of glucose to purple hexagons to glucose 6-phosphate upon entering the cell. This acts to trap glucose 6-phosphate inside the cell for later use. What does it mean to trap in this context? And what are the implications for further glucose into the cell? Well, as glucose is entering the cell, let's say we didn't do this reaction yet. Okay, so ignore the ATP to ADP. As glucose is entering in the cell, we'll end up with a lot of glucose inside the cell. Duh, right? As it's entering, eventually we'll even out. Now, once the concentrations of glucose outside and inside are equal, the spontaneous flow of glucose is neither direction, right? Neither side is favored. In other words, the chances that the glucose leaves the cell is equal to the chances of it coming inside the cell. And when that happens, the cell is not happy. Basically, if a cell is at equilibrium with an environment, that thing's dead. Okay. So we don't want that to happen. We want glucose to keep coming inside of the cell for us to use, for the cells to use. So we need to find a way to trap it. We need to find a way to keep it inside the cell and have more glucose spontaneously come in. So what we do is we react it with ATP. So we have glucose plus ATP going to glucose 6-phosphate plus ADP. Because now what we've done is we've artificially lowered the glucose concentration in the cell, right? By getting rid of all that glucose and converting it into glucose 6-phosphate, we've kept the concentration of glucose inside the cell very low. Which means, right, remember earlier we talked about a reaction. If I keep the product concentration low, I keep the favorability of my forward reaction high, right? High favorability. So I'm keeping the flow. Oh my God, can I draw this arrow correctly, please? Thank you. The flow of glucose from outside to inside still very highly favorable. Okay, but now I have this issue where I have an abundance. So lots of glucose 6-phosphate in my cell. And so we have high concentration of glucose 6-phosphate inside the cell, very low concentration. So my spontaneous flow of glucose 6-phosphate is inside to outside. It does want to flow out. But remember, spontaneity does not equal rate. Something can be spontaneous and take forever to take place. This is one of those cases. The glucose 6-phosphate spontaneously will go from inside to outside. It'll go from high concentration to low concentration. But the rate is so unbearably slow that it's irrelevant for biological conditions. Because glucose 6-phosphate has a little trick up its sleeve. If we look at what glucose 6-phosphate looks like, here's our glucose molecule. We have CH2. And a big ol' negatively charged phosphate group attached to it. And remember, he asked you to memorize that ions have very low membrane permeability. So even though the delta G says, oh yeah, or the spontaneity says that we'll go inside to outside for the glucose 6-phosphate, this will happen at such slow rates because of this negative charge on our glucose 6-phosphate that it's irrelevant to the cell. Okay. So we've kept glucose coming into the cell, and we've prevented our glucose 6-phosphate from leaving the cell because this phosphate group makes this molecule charged, and it won't be able to pass through the very hydrophobic core of the cell membrane. So now we just got to find the answer choice that corresponds to that. And I made it easy on you guys. A, trap refers to the addition of a negatively charged phosphate group that results in a molecule with a very low membrane permeability. Charge, very low membrane permeability. And the spontaneous flow of glucose remains outside to inside. There's our answer. B says trap refers to the addition of a positively charged phosphate group. Phosphate groups are not positively charged, so it's wrong. C, trap refers to the addition of a negatively charged phosphate group that results in a molecule with a very low membrane permeability. Okay, that's true. The spontaneous flow of glucose changes to inside to outside. Nope, there's no change. That's wrong. We want the spontaneous flow of glucose to remain outside to inside. D, trap refers to the addition of a negatively charged phosphate group that results in a molecule with a very high membrane permeability. Nope. Ions do not have high membrane permeability. E, trap refers to the addition of a neutral but polar phosphate group. Nope. They're not neutral. In order to get phosphate groups to be neutral, you have to have a seriously low pH. So no. And the polar phosphate group would not result in a molecule with a very high membrane permeability. Glucose already has a pretty low membrane permeability, hence why it has a channel. And so adding another, even if it were true that phosphate was polar but neutral, sticking a polar thing onto an already polar molecule would make an even larger polar molecule, which would make it even less permeable to the membrane, not making it highly permeable. So there's multiple issues wrong with E. Okay, so that is my practice exam. Congratulations for making it through if you did. My goal was that my practice exam will be harder than the actual exam. We'll see what happens. I guess only time will tell. I wish you guys the best of luck with your studying. Make sure to get some sleep. Don't be like me and be up till 2 a.m. I'll be up later than this, actually. I'll go drink some more coffee. I don't know. But yeah, so my exam is designed to be harder than the actual exam, but not so hard that it's destroying your confidence. So if you are finding that it is destroying your confidence, I'd recommend not doing them. Okay, because they're not intended to destroy your confidence. Okay, that is not my goal. Okay, but best of luck studying. Get some sleep. Make sure to eat. You guys are going to crush it. And I'll see you guys Friday. Or maybe not. I don't know. Hard to say.